All right. If you thought the last lecture was fun, just wait for this one. This one's just hold on. Here we go. We're going to move into how B cells generate diversity. Honestly, this is one of my most fun ones to talk about. It is going to be likely one of the most confusing to go through, because this is really different than anything you've ever learned before, because nowhere else in no other cell in the human body does the process we're going to go through occur except for in immune system cells. So the only way you would know about this process is if you had taken immunology. This is where you're going to learn it. And it's a lot of genetics, but it's completely different than the genetics you've learned in the past. So just, bear with me and pause, stop, review, look up other sources online, whatever you need to do to get through this. But hopefully by the end, you have a pretty good grasp on how diversity is generated in B cells. So we did in the last lecture talk about antibodies and immunoglobulin and how there's a huge diversity, right? For every possible epitope that is out there in the world, we're supposed to be able to be prepared to generate an antibody to match that. Well, that's like billions and billions and billions of different proteins we'd have to make. And we don't have that much DNA. And so immunoglobulin genes are set up differently than any other gene in the body. And those immunoglobulin genes are only turned on in developing B cells, and you'll see in developing T cells. So in those developing lymphocytes. So it's really pretty crazy. So in all cells, except for B cells in this case, because we're only looking at B cells, the immunoglobulin genes are in a fragmented form. They're not set up where this section of the chromosome codes for this gene and it's just transcribed and translated as is, but rather they're fragmented into what we call segments. And so instead of having that complete gene just in a package, the heavy and light chains of immunoglobulins are divided up into pieces or segments, and then they're still sequentially aligned on the chromosome, but different segments can be pulled out. It's like being a DJ and you have all these different segments of music, and instead, like everybody else just listens to a song and then goes to the next song and listens to it full, the DJ will mix and match and pull and combine to make a really cool product, and every product that DJ makes is just slightly different. And so that's what it's like to be generating diversity in B cells. So when these arrangements then are made, eventually there's going to be a complete heavy chain transcribed and a complete light chain transcribed, and then those heavy and light chains can join and be exported to the B cell surface. So let's look at what those gene segments look like. They're going to be located so if we're just talking about immunoglobulin now, and we're looking at the heavy and light chains that are going to be located on chromosomes 22, 2, and 14 in a section of those chromosomes. And so the heavy chain locus is going to be found on chromosome 14, and then we have two different and that's going to be all 5 isotypes. So that's going to include the gammas, the epsilons, the mu's, all of them are going to be on chromosome 20 or 14. Where the light chains are actually going to be encoded on 2 different chromosomes. The kappa ones are on chromosome 2, the lambda ones are on 22. You don't need to memorize this, but it's just nice to know. So here is an image that shows those 3 sections of the chromosome and what those segments look like. And you can see that with let's look at the heavy chain on chromosome 14 to begin with, down here on the bottom. So chromosome 14, we have a lot of different segments, and this is where our letters are going to come into play. So remember that light, L stands for light, oh, the lambda, sorry, this is lambda. I'm already messing it up. Oh, it's gonna be a long one, right?
Okay, so we have no, that's L for light. Sorry. This is lambda up here, kappa. This is the heavy chain. It's my whole reason for trying to go slow. Okay, sorry about all that. So we have our variable region of the heavy chain, our variable region of the heavy chain. This L stands for liter. Sorry, that's where I got thrown off. So L is liter there. It's just it's a liter a peptide that's made. So just you can kind of just ignore that. And then you have your liter 2 and liter 3. But so we have our variable heavy, and our variable heavy, but there's like 40 some of them. So there's a lot of different options for that variable heavy segment that can be used. Maybe this one is used, maybe this one is used. Then when we look at the that's just the variable portion. Then there's also going to be two other portions, the diversity and the joining, that are part of that whole variable region of the heavy chain, this big. Then we have our constant regions, which that's where our mu's and our epsilons and delta heavy chains are going to be. Now, if we look at the light chain, it's a much smaller chain, but there's still a lot of segments. And so for the variable region of the lambda up here or kappa, you have around 30 to 35, depending on which one you're looking at, and they're different segments. They don't have a diversity segment though, they only have a joining. So they just have the joining and the variable. Okay, so let's look at this before I make it even more confusing than I already did. Okay, so had I put this up here, we would have saw that liter piece. Okay, so the different gene segments then are going to be flanked by the liter peptide, and then those variable regions and constant regions of all of the 3 different chains. Okay, so the variable region then is going to be made up of the variable light chain plus the variable heavy chain. We saw that in the first lecture. And those segments must be selected from the different gene segments that are available. And they're rearranged, and then that will create an exon that is eventually going to be transcribed and then translated. Within the light chain variable region, there's 2 different segments, 2 different segment types. There is the variable. That's what we see in the red. We see the joining. And those are the 2 that make up. And so that we see that with both the light chains, variable in red, joining in yellow. Now, the heavy chain also has the variable in red, also has the joining in yellow, but it has an additional segment called the diversity. And so that variable region consists of 3 different segments that get put together to create the variable region. And then these constant regions come in later. Okay, let's look at how that happens. And I love these next images. I think they do a really nice job of explaining. And again, keep your DJ analogy in the back of your mind and thinking of these as like pieces of songs. So during B cell development, when a B cell goes through development, and we're going to look at that in a later chapter, but it's going to be spending a lot of its early time doing this genetic rearrangement. This form of genetic rearrangement, because it's so different from any other process, it has its own name, and it's called somatic recombination. So when those V and D and J segments are moved around and put together, that's called somatic recombination, and there's special enzymes that are involved in that process. So somatic recombination is a big vocabulary term from this chapter. It's the special recombination of these segments to make the variable region, and then eventually bring in the constant region. In the light chain,
there's just one set of recombination that occurs, and that's with the variable light and the joining light. So I love this picture because it shows us what, so there's a couple, let's look at just the light chain right now. So we're on this part of the picture, okay, just on the light chain. We have our germline DNA. So this is what was inherited. In that inherited form, on the chromosome, nothing magic has happened yet. And so we have our variable segments, we have our joining segments, and then our constant region that follows. Of these segments, we know there's like 60, whatever, or of the variable region, like 40 or so of the different segments that it could be selected from. Joining has a handful of them, constant will have a handful. And then this process of somatic recombination is what we'll select and move in and select and move in, and then eventually this will then be transcribed. When the DJ picks pieces of these songs and pieces of these songs and brings them together to make a really banger mix. I don't know if that's the right words to use. Then we get a functional song to dance to, or we get a functional protein. Now, heavy chain does the exact same process. There's just an additional recombination event that occurs where we have the variable one picked. So again, we have our germline DNA on this line. We have one round of somatic recombination where the D and the J join, which is kind of fun because it's the D to the J, kind of like our DJs doing the somatic recombination. And then we have a second somatic recombination event when we bring in the V. So the D and the J are joined, and then the V is brought in the second level. So a segment from D is joined with a segment from J, and then a segment from V is brought in to join the D and the J, and then we end up with a variable region that can be transcribed. Then we'll eventually be joined to a constant region to make the full peptide chain. So these are somatic recombination events. Two of them occur in within the variable region of the heavy chain. One of them occurs in the variable region of the light chain. Now, the particular segments of the V and D and J that are joined together are completely random. There is no rhyme or reason how things happen or why things happen. It's completely random. And the number of combinations, because of such a wide variety of segments to pick from, is essentially endless. There's billions, like you could never run out of options for combinations eventually, like across the whole repertoire. Okay, now a single combination event can run out, but then a new cell tries again. The enzymes that are responsible for somatic recombination are called recombination activating genes, or the enzymes, but they're coded for on the recombination gene, which is the reg genes. So we call them reg 1, reg 2. They're only involved in somatic recombination. So like that, somatic recombination. So only turned on in cells that do somatic recombination, and in our case, we're talking about B cells. So no other cell, we'll see T cells do it too, but no other cell at this point here is going to have reg genes turned on, only developing B cells. Now, each of those V and D and J segments in the variable region are going to be flanked by sequences that the reg enzymes recognize. And we call these recombination signal sequences, or RSSs. And so reg 1 and 2 proteins are these enzymes that will recognize those sequences and know where to bind. And so that's where the enzyme interacts. That's where that reg protein interacts, and will then loop the DNA together to bring sequences in juxtaposition so that they can be spliced together. And then eventually, and everything else is looped out and cut out. And so we see that happening in this image here on the bottom. We have our big string of DNA with all of our different sequence options on there for the variable region. And we have these sequences that are recognized in between. The reg enzymes will recognize those sequences, bring these close together, and then this is joined together and cut out. And then you end up with what is left, the splicing together. So the important thing to remember here is not so much the whole process of what actually happened or of the details of what happens, but rather reg enzymes are responsible for somatic recombination. And somatic recombination brings in different gene segments to make a wide variety of diversity of the variable region where we need to have lots of variability to recognize all the different possible antigen specificities out there. Okay, now there's going to also then be, that's not enough diversity because we still only have so many segments and there's only, you know, 1 billion or so opportunities, but there's more, right? So there's more diversity generated in the variable antigen binding region in that hyper variable region by way of something called junctional diversity. Junctional diversity is going to be done by enzymes that bring in and just start adding in random nucleotides. It's like, yeah, let's throw some nucleotides in here. We'll throw some nucleotides in here and just kind of mix it up a little bit. I know those nucleotides weren't coded in the mRNA, but who cares? We'll just throw some in. And so what that does then, those are called P and N nucleotides. It's because of the palindromic and I don't know what the other N stands for. Must be, I don't know. It doesn't matter. They're P and N, but they'll just add in extra nucleotides and make more diversity. And so we call this junctional diversity. And it's going to increase the diversity within the amino acid sequence. Okay. Let's see here. What did I miss on this one? Yeah. Oh, TDT. That's an enzyme that's going to be doing some of that work. Okay. So additional. So you have somatic recombination. That's going to be one level of diversity. And then you have junctional diversity, which happens after the protein's been transcribed and translated to add in additional nucleotides or before transcription. Okay, not translation. The rearrangement of those segments of the variable and the diversity and the joining segments will create, we're looking at the light chain here, I think. Oh no, we're looking at the heavy chain. We'll create that variable region. I knew it was the heavy chain because of the J and the D and the V. There's 3 of them for the heavy chain. Also, the constant regions then are mu, our delta, our gamma. And so once we have those V and D and J segments in juxtaposition, then transcription can occur. mRNA will be created from transcription and then is going to be spliced and processed just like any mRNA is spliced and processed. That situation is no different, but we'll bring in our constant regions. So then we can have mu, or we have mu and we have delta as our first 2 that are closest. And so those are going to be the 2 first that are going to be made because they'll be brought in first. And we'll talk about that in just a little bit more detail later. Okay. Here's an image now that will kind of bring all of that information together, looking at both the light chain and the heavy chain side by side. So let's start with the light chain. It's kind of a really great review slide. We have our germline DNA up here. So our light chain is going to have variable segments, joining segments, and constant segments. And it can be kappa or lambda. We don't know. It's going to happen on both of those chromosomes. Our germline DNA for the heavy chain will have our variable segments, our joining segments, but also our diversity segments, and then all of our constant regions, okay, with mu being the closest. We have our first round of somatic recombination occur, where in the heavy chain, we're going to have our D and J segments joined. So then we'll have a second round of recombination occur, where the light chain, well, the light chain is in the first one, the J and the V. And then in the heavy chain, we're going to join the V to the D and the J. Okay, so now we have rearranged DNA for the variable region of both the constant and heavy, both the heavy and the light chains. Transcription of that is going to occur. There's a primary mRNA transcript made. Then there's going to be some splicing where we bring in a constant region. Bring in one of these constant regions. It's going to be one of these first ones. And then the mRNA is going to be translated into our protein. So we have our variable region of the light chain plus our constant region of the light chain made in this first diagram. And then in the heavy chain, we have our variable region of the heavy chain, and then all of our constant regions of the heavy chain brought in. So it's a really great slide. I really take some time on this one if you're having difficulties understanding. But again, using that DJ analogy can probably help a little bit. Now, DNA, okay, sorry. Developing B cells are going to use an alternative mRNA splicing event to make IgM and IgD. I think my picture might be over that, but it says IgM and IgD. Because both mu and delta constant regions are closest to the variable segments, those are going to be the first two made, and that alternative splicing is just going to bring one or the other in. When a B cell that is circulating, but it has not encountered antigen yet, we call that a naive B cell, and we're going to use that term a billion times. Naive B cells. That means that they have immunoglobulin on their surface, but they have never been exposed to their specific antigen. So they're naive. They will have both IgM and IgD on their surface because of this alternative mRNA splicing. The transcript has been made, but then it's going to be altered and altered and altered to make either IgM or IgD constant regions because of the splicing events. So this process then of rearrangement, like you might imagine, is very tightly controlled. These segments are not just these somatic recombination events don't just happen willy-nilly. It's only going to happen at one time, and only one event can happen, and this is going to lead to the production of only one light chain and one type of heavy chain. You won't have 72 different chains made here of the heavy and 72 of the light. That'd be a mess. And so B cells have a process called allelic exclusion, meaning that they are going to only recognize one type of antigen. Whether they're IgM or IgD, it doesn't matter because their variable regions are the same. And so that's called allelic exclusion, and that will ensure that a B cell produces IgM and IgD, but only has one antigen specificity because the variable region does not change. And so every B cell is going to have one single specificity. Okay, now this is where that whole concept of immunoglobulin versus antibody is brought up. We can have two different forms of immunoglobulin. We can have a membrane-bound immunoglobulin, which means it's attached to the surface of a B cell.
So this can also then be called a B cell receptor. And lots of times you're going to see B cell receptor abbreviated as BCR. B cell receptor. So when this B cell first makes IgM and IgD, it's not secreted. It will enter the endoplasmic reticulum and be shuffled out to the surface, and it will maintain a connection to the membrane. And that is how they stay attached. But now the B cell receptor actually includes another set of protein chains called Ig beta, Ig alpha. They're important because their tail that extends through the membrane is longer than the immunoglobulin, and so it can signal better. Signaling occurs through Ig alpha, Ig beta. Ig alpha and Ig beta have no variability. They are the same sequence across all B cells because they're just regular coded on the genes, and they're just transcribed and translated like regular proteins. But they will join with immunoglobulin, transport to the surface of the B cell, and stay there, and together make the B cell receptor, these two together. And they are the antigen recognizing structure on naive B cells. So here's an image, a closer image of that. You can see that we still have our antigen binding sites within the variable region of the immunoglobulin. When these antigen binding sites are engaged, signals will be sent, but the cytoplasmic tails are too short. And so they can't interact with the cytoplasm receptors and everything down in the cytoplasm, but rather those messages will be transferred down through Ig alpha, Ig beta, and they can send the signals down to the cytoplasm to then turn on transcription factors to get things going. And so really Ig alpha and Ig beta's whole purpose is signal transduction. Again, these two proteins collectively are called the B cell receptor, and they're found on naive B cells. Now they can be IgM or they can be IgG. No, delta, IgD. Okay, so those are the B cell receptors, and it could be a mixture on the cell surface. Okay, here's our image again of the somatic hypermutation, or the somatic recombination events that occur. And we have our transmembrane IgM and our secreted IgM. This is also called antibody. AB stands for antibody. We don't really call it secreted IgM, we call it antibody. They're essentially going to be the same. It's just that there's going to be this terminus changes, and so then it will not be embedded into the membrane. Okay. And these are going to be made by plasma cells, and these are going to be on B cells, naive B cells actually. Okay, you heard me kind of slip up here and say somatic hypermutation because it's another process. So here's another major, so we had a somatic recombination where we had the V and D and J segments joined together for both the heavy and light chains, V and J on the light, and we call that somatic recombination. Once that's done and a B cell has made immunoglobulin, it's on the surface of the cell. And let's say that a B cell then receptor recognizes antigen and some signals get transduced down into the cytoplasm. Now the B cell is like turned on again, and we get to re-engage those reg enzymes, and we get to reevaluate that variable sequence. And that process is called somatic hypermutation.
because it's a lot of slight changes in the variable region of both the heavy and the light chains. The constant region is not affected at this point. And this is dependent on another special enzyme that's only turned on in B cells that have been activated, and that is AID, induced cytidine deaminase. This enzyme is going to provide a whole bunch of B cells now, it's called proliferation, and each of these B cells is going to have a slightly different type of immunoglobulin. And so they're going to be mutant. They're going to change ever so slightly from that initial immunoglobulin that was made. Some are going to have a higher affinity because these slight changes in that variable region might make the immunoglobulin bind stronger to the antigen. Some might not. Some might have worse because of the change. So those B cells then that have the best or the highest affinity immunoglobulin are going to compete and win because they'll bind stronger, and they are going to be preferentially selected for, meaning they're the ones that are going to be able to go on to the next step of adaptive immunity. And so as this response proceeds, the antibodies are just going to get better and better and better and progressively have a higher affinity. And we call this process affinity maturation, which is another good vocabulary word from this chapter. Okay, that's where we're going to stop as I say that, we're going to stop there and pick up on our last piece of differentiation in the third section.